the University of Arizona Bio5 Institute, we bring together hundreds of multifaceted experts that include world-class bioscientists, engineers, physicians, and computational researchers. This team science approach is designed to ignite creative solutions to the many complex biological challenges facing our families and communities, and has resulted in disease prevention strategies, promising new therapies, innovative diagnostics and devices, and improved food sustainability.
Hello and welcome to another episode of Science Talks, a conversation hosted by the University of Arizona's Bio5 Institute. My name is Lisa Romero. Neurological diseases are disorders that affect the brain and the nervous system and include Parkinson's disease, dementia, and Alzheimer's disease, among others. These diseases affect hundreds of millions of people around the world. Researchers are working diligently to better understand these diseases as new approaches to prevention, diagnosis, and treatment is paramount. Today, we're joined by a trained physician and scientist, Dr. Lalitha Madhavan. Dr. Madhavan is an Associate Professor of Neurology and also a member of the Evelyn F. Mcknight Brain Institute and the Bio5 Institute here at the University of Arizona. Her research merges the fields of neuroscience and stem cell biology to understand how the brain works and contribute towards the development of treatments for disorders such as Parkinson's disease. She's also a recent 2022 University of Arizona Women of Impact awardee. Dr. Madhavan, thank you so much for joining us today. Well, I'm really excited to be joined today by Dr. Lalitha Madhavan. I am a huge fan of your work and how meaningful it is to a lot of areas of health that are important to me and a lot of other people. But mostly I'm really honored to be in the presence of one of the 2022 Research Innovation and Impact Women of Impact Award winners. So I want to congratulate you on that honor. That's pretty incredible and well deserved.
Yeah, thanks, Lisa. Thanks for the opportunity, and it's just fantastic to be here.
Oh, good. Well, I'm so glad you're here as well. I want to get to your work and talk to you about a lot of things, but I think let's step back for a minute. I'd love to hear a little bit more about your background, how you are a physician researcher, which is incredibly noble and difficult, and there's not a lot of people that manage to do one or the other, much less both, and merge them into a successful career. So I can't wait to hear more about that. But I think you went to medical school in India, right? Were you raised in India?
Yeah, I was raised in India, and I did go to medical school there. So, I became interested in the health sciences very early. As a child, I always wanted to do something related to the health sciences, but my first love and my first stop was really medicine. I was lucky enough to get into medical school. I made it through it, and then I was just kind of going along, trying to build my career as a physician. But just as things happen in life, very often things took a little turn, and I ultimately landed up in research. So predominantly now I engage in research. The backstory of all of this is really thinking back about things. As I was in the clinical arena for those few years, it became clear to me, a few things became clear to me actually. One was that, especially in terms of neurological disorders, we didn't understand these disorders very well at all. And that was a big part of the problem in terms of developing treatments for these disorders. And, there aren't really treatments that can address these disorders meaningfully, that can stop the progression of these disorders. there are very few options actually for physicians in the clinic to provide to patients that can really help them. And so as these sort of realizations were coming upon me, when in my clinical years, I had moved abroad, and I got a chance to do research, something that I hadn't had the chance to engage in during my medical school years. And then everything started to click. It started to seem to me that in order to make advances in brain disorders, advances in basic research were important. And so things took a turn, and I started to delve into research and went on the long road of the PhD and the postdoc and all of that. And, I guess, that's just how it all started.
Here you are after all that, right? That's an amazing story. I mean, I think that, we at Bio5 are lucky to have, a handful, maybe a double handful, but not many, of physician researchers that have done kind of taken both of those journeys, either, like you said, started off with one and, kind of somehow got into the other, or somehow in parallel, it evolved. But it's always sort of that same narrative that you shared is just this whole different mindset that brings when you are combining the training and the, I guess, lens of both backgrounds into research outcomes now. So I want to talk about your work today. So I mean, you start doing research, you find that it really is a fit for what you want to pursue. How do you just start out on the path of neurological diseases, and in particular for you, I think Parkinson's is a primary focus, although I know there's implications for other brain diseases as well in your work. But I mean, how did that happen, and sort of, was there a personal interest in that? I mean, it just sort of evolved.
Yeah, during my PhD, I was lucky because just around the time that I joined the program, there were two researchers who joined as faculty in my department, who were pioneers in the area of stem cell biology and cell transplantation. And they came to the department, and I was lucky enough to be able to impress them and join their lab as a student. When I started to get exposed to this field of stem cells, it really became obvious to me, the sophisticated and cutting edge and almost the idea that this research was at the frontiers of science, and there was just so much potential and so many avenues through which there could be solutions for some of those problems and some of those issues that I had been thinking about, especially during my clinical years, and then moving into that whole period of trying to swerve into the research side of things. And so that's where my journey with stem cells started. Then when I went on to do my postdoc, I joined a lab that had expertise in Parkinson's disease. My mentor was again one of a very, very well-known scientists in the Parkinson's disease area. And so there came the interest there. So my job there really was to kind of apply and bring my stem cell expertise to bear upon those issues relating to Parkinson's disease in that lab, and I've just followed that path since.
Yeah, that's amazing. I mean, I think, for us non-scientists out there, you talked a little bit about this already, but it's, I think, very hard for people to understand with brain diseases becoming so prevalent and so damaging to virtually every family at some point, in some way or another. And I think, I hear a lot, even friends of mine that are not really, around academia or science a whole lot, and just like, how can we not be further along in understanding what causes dementia, what causes Parkinson, and like how is it possible with all the technology and this, you know, science that we have emerging all the time, how can we be still so far behind? And I try to explain in my very, very non-scientific way, but just because I am lucky enough to be able to talk to so many of you, because it's the brain, and it's very difficult to study, a brain in a live person, and just it's difficult, it's complex, and so I would love for you to talk about how, and I mean, I guess in parallel to that, I think a little is understood, or maybe some is misunderstood about stem cell biotechnology.
Mhm.
And I think maybe some people have a reaction to that without ever understanding, the incredibly positive benefits of this type of work. So talk about, how stem cell biotechnology, is allowing, ability to potentially develop, these biomarkers for earlier diagnosis, perhaps novel treatments and therapies. Talk more about where you are with that in your work.
Yeah, sure. So, let me take a step back and just kind of talk about what stem cells are and what these things, just to give a perspective,
That's
on
great.
on their
Yeah.
potential. So, like you said, the brain is one of the most, highly evolved and complex organs that really makes us who we are and defines what we do in many ways. But it's also a very remarkable organ in the sense that it is learning, it is changing, and it's adapting throughout our lifespans. And that adaptation, the scientific term for that kind of adaptation is plasticity. And I think this terminology probably has been thrown around a lot, but plasticity essentially means adaptation, and stem cells are really at the core of this adaptive process because they have the ability to divide and generate new cells and replenish tissues in a way that other cells cannot. And so these cells really remain as sort of reservoirs of plasticity in our brains throughout life. Reservoirs that can be used to support brain health and maintain brain resiliency in a way. But also, we now know that we can sort of grow these stem cells, in the laboratory. We can study them. We can sort of use them to sort of, I would say, test drugs and sort of develop treatments for disorders where certain cells have died. Because like I said, stem cells can become other cells, other different type of mature cells. And so, we've developed now these techniques where we can drive these stem cells in the laboratory to become these cells that are dying off in brains in particular disorders. And, there's this idea of trying to replace cells, right, back into the brain, in brains of patients who have lost some of these cell types. So those sorts of possibilities are there with stem cells. And so, that's been really exciting. This is not possible to do with other cell types in the body or other types of technologies.
That's really interesting. And, I know at U of A, it's a very unique space where we have a lot of, really world-renowned, recognized scientists, researchers working in, the field of what is normal aging, what is aging, what is normal aging versus diseased aging, disease as we age. And I think, probably this kind of thinking would be useful in helping discern those two things as well. Is that right?
Yeah, absolutely. That's correct. I actually work very closely with the aging biologists here at the UFA. I'm part of the Mcknight Brain Institute run by Dr. Carol Barnes. And, it's just been fantastic, being part of that environment too, because when I initially came here, I wasn't into the aging field as much. But, just because aging is just so intrinsic to chronic neurological diseases like Parkinson's, because we know, that Parkinson's occurs in people, has more chance to occur in people as they age. Not all people who age are going to get Parkinson's, but generally Parkinson's occurs in people who are older. And so that sort of connection between aging is very interesting. So to understand what is normal aging, and then what differentiates normal aging from pathological aging or disease is really important to understand. And so we kind of straddle that side of the work too. So stem cells are very useful in studying those kind of issues too, because although they are a fountain of youth, they are also affected by aging to some extent. And so they can be reporters and tell us a lot about aging, actually, and what's going on there. So we have used stem cell as a tool to study aging from that point of view.
Well, I'm not going to lie. I wish that you all would know everything there is to know about this before I'm as old as I am already, as I'm sure you can relate to. But it gives me so much hope for the next generation, my children, our grandchildren, and it's just so exciting. But when we're talking about stem cells and looking at them again in regards to aging versus disease, and how they can re-energize, how they die, how they whatever the process becomes, can you basically look at individuals in their specific set of stem cells and study the causes of again particular disease, or just look at it and say this is a normal aging process?
Yeah, no, that's really a complex issue and difficult to study. But we are able to study actually diseases from a very individualistic point of view using stem cells these days. So this really goes back to this technology called induced pluripotent stem cell technology. It's something that I've established in my own laboratory since joining the University of Arizona. But essentially, this technology was given the scientists who developed this technology was given the Nobel Award in 2012. And this technology really is about isolating some sort of adult cell, like a skin cell or a blood cell from an individual, either diseased or normal. And then you could, through a series of research manipulations, you could sort of drive these cells to become embryonic-like cells. So embryonic stem cells are the ones that give rise to all other cells in the body. And so now, from an adult cell, you've sort of given rise to this embryonic like stem cell without delving into embryos, which is, of course, there's a lot of ethical issues associated with that, and feelings and emotions run high in that area. And so here, there's a different way to generate embryonic cells. And then once you have embryonic cells, they can be grown technically forever. They can be banked in laboratories. And then you can drive these embryonic cells from there into other cells, like certain types of neurons, for example, neural cells that one would like to study that are, for example, of importance in Parkinson's disease, which is what we do. And so now we are able to access brain tissues in a way from living individuals that we haven't been able to access before. Because like you mentioned, it's not possible to study brain tissues from living individuals. You can't take a brain biopsy from somebody like you could take a little skin biopsy, or if there was a tumor or something, take a little and look at what cells are doing. And so this really gives us this indirect way of generating brain tissues from different individuals. And the unique and really exciting thing about this whole technology is that what we're finding is that these cells can actually be reporters of sort of what's happening from individual to individual. So, for example, cells from somebody who with Parkinson's disease behave differently in culture compared to somebody who doesn't have Parkinson's disease. And even between individuals who have Parkinson's disease, each of them has a different, their cells behave differently even within the group. So we all know that everybody's disease is not the same. Everybody expresses the disease differently. And for example, that's important because the same sort of medications that we think may work for everybody may work well for some and then not so well for others. And so this sort of studying the disease individual by individual really has the advantages that you can be very precise in sort of what you learn about individual variations, and you can understand the disease better, first of all, what the variations are, and then you can test drugs on some of these cells and see, maybe the cells works on cells from individual A and individual B, but then the drug doesn't work on cells from individual C. And so this way you can really sort of be a little bit more focused and strategic in terms of how therapeutics are developed. And I think the broad terminology for this these days is precision medicine. And so this really allows, this technology really allows for precision medicine to sort of be applied in a way that, again, we haven't been able to do so in the past.
It's so exciting. I'm thinking about I know another passion of yours is mentorship and making sure that you're passing on this type of experience to others. And I'm thinking of these lucky students that are in your classroom, if you're teaching in your lab, and being able to work side by side as all this really evolves and unfolds. I know that's a passion of yours, and talk to me a little bit about how you involve the students in this as sort of you go along.
Yeah, I am passionate about working. How should I put it? I think I'm passionate about working with a diverse slate of students because I really think that diverse opinions matter for anything, and it propels creativity, and it also tremendously helps students in gaining confidence and a worldview. And so I enjoy interacting with students at different levels from all kinds of backgrounds. And, my basic philosophy is that it's not really important where the students are academically or, what they've learned really in the past. It's more what they want to do and the motivation and the interest and the drive that they show to want to get somewhere or want to learn in the lab or in the class, wherever that might be. And so, it's just been fabulous just interacting with such bright and engaging and lovely individuals. I feel so lucky. From that point of view, I love the job of a professor because that allows me to do something like this. Especially undergraduates. I love interacting with undergraduates because I feel that it's a very important part, a time period in the trajectory when students are trying to make decisions on to where they really want to go. And I feel like I can make meaningful impact at that time. And also they're very fresh, they're not afraid to ask questions. They bring a lot of new ideas onto the table. As we get older, we develop a few more inhibitions and we're a little bit more worried about what other people might think of us. I find undergraduates mostly, are a lot more freer in how they sort of go about things. And sometimes they ask those so-called dumb questions that are really very important. And so, it's been fun interacting with them, and I usually tend to give them independent little mini projects as they learn and do things so they can really try to understand the ropes of research and understand, from the beginning to end what the research process means and also what it means to clinical medicine. How does that, because I really look, as you mentioned and I'd mentioned before, taking care of the patient, I truly feel involves both arms, the basic side as well as the clinical side, and they need to go hand in hand and they need to talk to each other. And so if I can convey that to the student or whoever, people that I mentor, at least I try to convey that. I think that's a very important message that I can give to them.
Yeah, and I mean, I think, it's something I love about being involved with Bio5 too, because I mean, two things you mentioned, this concept of diverse thinking and diverse backgrounds, and we mean that, very broadly in every possible way. Just diversity in general is so much a part of what makes, Bio5 a special place, and the University of Arizona, I think, really does a good job of actually, walking the walk of that, not just saying that that's what we do, but as well as bringing students in so early into, we have our high school internship program, Keys, and the same thing you described. It's just amazing to see these minds come in and, just deer in the headlights, but then, they get in the labs, and they get in a lab like yours, and just the inspiration and imagination and creativity, and like you said, lack of a filter come out. It's really exciting. But it takes special people like you that are willing to put that time in and see really the benefit, both from their perspective, but also from your own. And it's really admirable. I love that.
Yeah, well, that's nice of you to say, Lisa, but it's really a two-way process. I mean, the students teach me so much along. So I'm better because of the students. So, that's something that's what I'd like to say from my side.
That's the best kind of relationship, right? It's two-way benefit for sure. Well, I know you're a busy person. What do you do in any free time that you might have? Do you have any hobbies, or what is your favorite thing to do if you're not in your lab or not teaching?
Yeah, I mean, I don't get enough time to do it. I wish I had more time to do things like that. But I love the outdoors, and Tucson is so beautiful. That's something that attracted me also to come to Tucson. In 10 minutes, you can be out in the wilderness, walking on some beautiful trail somewhere. I love hiking, so I try to get out there and do that as often as I can. My husband is an avid hiker too, so we both like doing that together. And I like listening to music and reading. All kinds of variety of things. There's no specific whatever, but yeah, those are a few interests that I try to kind of push along on the side.
Relaxing the brain a little bit, and just being immersed in nature, music, whatever, to kind of stop everything for a little bit, right? Yeah. Sure. Well, thank you so much for being with us today and telling us about your work. I'm really excited for what's ahead. I hope you'll come back and visit us again, and just talk more about what's coming up. And thank you for all you're doing. I think that the unique space of your work, is going to make a big difference in a lot of people's lives. So thank you for that.
Yeah, thanks so much, Lisa. It was really enjoyable talking with you.
You too. Take care.
All right. Yeah.
All right. Bye-bye.
Bye.
Thanks to our listeners for tuning in to another episode of Science Talks. Continue the conversation with us next time as we learn more about the amazing science happening at the University of Arizona's Bio5 Institute.